for decades, oncologists basically chased a white whale. Like, there was this one specific cancer mutation, a microscopic anomaly that researchers openly called the holy grail of oncology. Or, I mean, in their darker moments, they just called it the undruggable target.
Right, the completely undruggable target.
Exactly. And then, after years of failure, science finally cracked it. We built the molecular harpoon, we hit the target, but here is the massive, incredibly frustrating plot twist. For some cancers, the tumors just, they simply shrug it off and keep right on growing.
It really is. I mean, it's one of the most fascinating biological paradoxes we face today. You hit a tumor with a molecularly perfect drug-like, a drug specifically designed for its exact genetic vulnerability. And the tumor doesn't just, endure it. It actively rewrites its own biology to survive.
And that paradox, that is our mission today. We are exploring the literal physical map of how a tumor outsmarts our most advanced therapies.
Yeah, it's a wild map.
It really is. So we're pulling insights from a landmark paper in the journal Cancer Cell by White, Campbell, and Sansom. And they're unpacking this just incredible study by Alonso and colleagues. And whether you are, a biology enthusiast tracking the future of personalized medicine, or you're just captivated by, like, the ultimate evolutionary chess match, this deep dive is going to completely shatter the old textbook view of cancer.
Absolutely, because we are moving way past the idea of a tumor as just, a chaotic lump of dividing cells.
It's a highly organized, shape-shifting ecosystem. So let's dive straight into the deep end here. What exactly is this undruggable target, and why were we so certain we finally had it cornered?
Well, the target is a mutated protein called KRAS. So to visualize this, imagine KRAS as a vital traffic light inside your cell.
Okay, traffic light.
Yeah. Its normal job is to basically relay signals from the outside of the cell down to the nucleus, flipping green to tell the cell to multiply and red to tell it to stop.
Makes sense.
But when KRAS mutates, and specifically we're talking about a mutation called G12C, the mechanism just completely breaks. The light gets permanently jammed on green.
So the cell just divides endlessly.
Exactly. Endlessly. And for the longest time, drug designers just couldn't stop it because the physical shape of this mutant KRAS protein is it's incredibly smooth.
Like smooth physically?
Yeah, structurally. There were no pockets to attach a drug to. It was honestly like trying to get a handhold on a completely flat, frictionless wall.
Wow. But science eventually did find a handhold, right? I mean, how did we manage to grip a frictionless wall?
Well, researchers made this brilliant discovery about the protein's behavior. Even when KRAS is stuck on the green signal, it isn't completely static. It briefly, almost microscopically, sort of cycles into a resting off state.
Oh, like it takes a breath.
Exactly. Yeah. That fleeting moment was the vulnerability. So we developed what are called off-state binding KRAS G12C inhibitors.
Okay, that's a mouthful.
It is, yeah. But basically, these drugs patiently wait for the protein to take that breath, and they slip into a newly discovered tiny pocket that only appears in that off state, and they permanently lock the traffic light on red.
That is genius.
It was a monumental breakthrough. I mean, it opened the door for even more advanced therapies that are in trials right now targeting, multiple states of the protein.
Right, so we found the key, we slipped it into the lock, we shut off the engine. Why on earth isn't this the ultimate cure for everyone with this exact mutation?
Because the environment surrounding that engine dictates pretty much everything. If you have lung cancer driven by this exact G12C mutation, these drugs often work remarkably well. The tumors shrink.
Okay.
But if you have colorectal cancer with the identical genetic mutation, the clinical response is, well, it's incredibly poor. The colorectal tumor just immediately resists the drug.
Wait, hold on. If it's the exact same broken traffic light, why does the address of the cell-like lung versus colon, why does that make such a massive difference?
It really comes down to the biological wiring of the tissue itself. So in colorectal cancer, when you shut down that KRAS protein, the cell experiences this sudden massive drop in growth signals.
It thinks it's starving.
Exactly, it thinks it's dying. So it immediately triggers a backup generator. It rapidly activates something called the EGFR signaling pathway.
EGFR?
Yeah. It's a network of receptors on the surface of the cell that acts kind of like a giant antenna, pulling in new growth signals from the surrounding environment to completely bypass the blocked KRAS.
Oh, I see. That makes me think of like a corrupted electrical grid.
Oh, I like that.
Yeah, so you managed to cut the main power line to a villain's headquarters. But the second the lights flicker, this automatic relay switches on, pulling power from a totally different municipal grid, the EGFR pathway, and the headquarters just stays online.
That captures the dynamic perfectly. And the medical community recognized this backup system. Doctors thought, well, okay, we'll just cut both lines.
Right, logical.
So they started treating colorectal cancer patients with a combination of drugs, one to lock KRAS and another to block the EGFR antenna.
Cut the main power, cut the backup generator. I mean, that should be game over for the tumor.
It really should be. But clinical trials revealed a pretty harsh reality. Even with both of those pathways blocked, patient responses were highly variable. I mean, some tumors shrank briefly, some barely responded, and almost all of them eventually grew back.
Unbelievable.
The tumor was finding a third way to survive. And uncovering that hidden survival strategy, that was the core mystery that the Alonso study set out to solve.
Okay, let's just pause real quick and map out what we've covered so far. We started with KRAS, this microscopic growth switch jammed in the on position. We developed a drug to sneak in and lock it off. But in colorectal cancer, shutting off that switch just forces the tumor to activate a backup antenna, EGFR. And even when we destroy the backup, the cancer still survives. Which leaves us with a massive question for you listening. If you cut off a cell's only known food sources and growth signals, what drastic measures must it take to avoid starving to death?
Yeah, and to answer that, we have to look past the genetic hardware. The Alonso study utilized this really fascinating combination of technologies. They didn't just read the DNA of the tumor. They used exome sequencing combined with spatial transcriptomics on patient biopsies.
And these biopsies were taken when?
Before treatment, during treatment, and then after the cancer became resistant.
Okay, let's translate those terms for a second, because I know the distinction is crucial here. Exome sequencing, that's looking at the blueprint, right? Checking if the tumor evolved a brand new genetic mutation to just physically block the drug.
Exactly, hardware changes.
But spatial transcriptomics, that sounds like a completely different approach.
Oh, it is a revolutionary leap.
Yeah.
So traditional genetic sequencing is kind of like taking a complex piece of tissue, throwing it into a blender, and analyzing the smoothie.
Right. You know all the ingredients in the cup, but you have zero idea how they were arranged.
Precisely. But spatial transcriptomics allows us to look at the intact fruit tart, so to speak. It doesn't just tell us which genes are turned on, it tells us the exact geographical coordinates of those actor genes within the physical architecture of the tumor.
Wow.
Yeah, you can see precisely which cellular neighborhoods are doing what.
And the findings from that spatial map completely upended the old theories, didn't they? The tumor doesn't just acquire a random DNA mutation to beat the drug. It completely alters its fundamental identity. It uses something called cellular plasticity.
Yes. Plasticity is basically the ability of a cell to change its state without changing its underlying DNA. It alters the software, not the hardware.
That is wild.
It is. And the researchers observed a literal timeline of this evasion, and it plays out in 3 distinct stages.
Right.
Let's look at the baseline first.
Okay.
So before the patient even takes the KRAS inhibitor, the colorectal tumor is heavily enriched in fetal, regenerative, and YAP epithelial cell states.
A fetal state. I mean, that implies the tumor is acting like embryonic tissue. It's not behaving like a mature adult colon cell at all. It's reverted to this pure construction mode, focused entirely on just rapid expansion and building tissue from scratch.
That is exactly the baseline. Then the patient takes the drug, the KRAS inhibitor hits the tumor and cuts off the growth signal. This is the acute phase.
Okay, stage 2.
Right. The tumor is in profound distress. It's essentially hitting the panic button.
But how does a cell in a panic attack physically alter itself?
Well, it suppresses all those fetal rapid construction markers. It realizes it just can't afford to build anymore. So instead, it radically shifts its identity into a canonical stem cell phenotype.
And that's characterized by a specific marker, right?
Yeah, a protein marker called LGR5. Specifically, it starts mimicking what's known as a Paneth cell.
Okay, I really want to dig into that because it's such a brilliant evolutionary trick. Because in a healthy human gut, Paneth cells have a super specific job. They live at the very bottom of the intestinal lining, and their entire purpose is to protect and nurture the delicate stem cells that regenerate our stomach lining. Right? They secrete all these protective proteins and antimicrobial shields. So the cancer cell is literally putting on the uniform of a specialized bodyguard cell.
It basically builds a survival bunker.
Yeah.
But it doesn't just hide quietly. Alongside this Paneth cell transition, an entirely new pro-inflammatory cell state emerges.
Oh, interesting.
Yeah, the tumor begins aggressively pumping out inflammatory signals into its surroundings.
Okay, but I have to ask about the energy economics of this. If the drug is shutting down the cell's main growth engine, how on earth does a poisoned, starving cell have the energy to completely rewrite its software, change into a bodyguard cell, and start blasting inflammatory sirens?
See, that is the genius of cellular plasticity.
Yeah.
It isn't generating new energy, it is completely reallocating its resources.
Oh, I see.
Yeah. It abandons the highly energy-intensive process of cell division and channels every remaining ounce of ATP, cellular energy into survival signaling and shifting its epigenetic state. It basically trades growth for endurance.
Wow. So we have the baseline construction phase and then the panic-induced bunker phase with the fire alarms blaring. What happens in the third stage, when the tumor finally figures out how to outsmart the drug and starts progressing again?
Well, the spatial map shows us that at progression, the tumor doesn't turn the alarms off. That pro-inflammatory state actively expands and solidifies, but simultaneously, the old fetal rapid growth state reemerges right alongside it.
Wait, really?
Yeah. The tumor manages to bring back its construction crew while maintaining the protective inflammatory bunker.
It learned how to do both at the same time? That's terrifying.
It is. And this behavior is incredibly consistent across different types of cancer models. The researchers point out that in genetically engineered mouse models, short-term inhibition of mutant KRAS forces the exact same shift.
From the regenerative state to that LGR5 positive stem state.
Exactly. And we've seen similar transitions in other models treated with entirely different drugs, like BRAF and EGFR inhibitors.
So it's a universal playbook.
Pretty much. It tells us that this shape-shifting ability isn't just a fluke. It's a fundamental, deeply hardwired survival mechanism embedded in the biology of intestinal cells.
Okay, let's do another quick synthesis for everyone tracking this. We now know that tumors survive our best drugs through cellular plasticity, the ability to rewrite their software. They start in a rapid growth fetal state. When poisoned, they hit the brakes, morph into protective stem-like cells, and trigger massive inflammation.
Right.
And then, once they adapt, they combine both the growth and the protection mechanisms to become virtually invincible. Which brings us to another really fascinating question, Fado. If a tumor is shifting into all these different identities, fetal, Paneth, inflammatory, is the tumor just a chaotic soup of confused cells, or is there a larger organized strategy at play?
And the answer there lies in the spatial transcriptomics we discussed earlier. When the researchers mapped these resistant cell states onto the physical tissue, they found something astonishing. The cells aren't randomly mixed like a soup at all.
Oh, they aren't?
No, they actually segregate into discrete, highly organized regions within the exact same lesion at the exact same time.
Let me get this straight. So within a single tiny tumor, you might have one microscopic neighborhood entirely populated by cells locked in the fetal construction state, bordering a completely distinct neighborhood of cells locked in the inflammatory bunker state.
Precisely. And this totally shatters the old idea that resistance is driven by one single rogue clone that mutates and just takes over the entire tumor mass. What we are actually looking at is a complex tissue microenvironment.
So the cancer is building distinct, specialized neighborhoods.
Yeah, neighborhoods that likely communicate and share resources.
This is where we have to look beyond just the cancer cells themselves, too, because the source material notes a highly complicating factor here. The targeted drugs are designed exclusively to hit the mutated epithelial cells. The cancer, right?
Right. The drug only cares about the mutation.
Yet the spatial map revealed that the massive inflammatory programs were activating in the stroma and the immune compartments as well.
Yes, and that is such a crucial distinction. For context, the stroma is the structural connective tissue surrounding the tumor, and the immune compartment obviously contains your body's own white blood cells.
Okay.
Traditionally, oncology treated these targeted therapies as if they were operating in a vacuum, only affecting the mutant cells. But this research proves that the cancer's survival strategy heavily manipulates its healthy neighbors.
Think of it like a corrupted ecosystem. Like the cancer cell isn't just a rogue weed growing quietly in a garden. The moment you spray that weed with pesticide, the weed starts pumping chemical distress signals into the soil.
That's a great way to put it.
Yeah, and those signals force the surrounding grass, the local earthworms, and the soil bacteria to completely alter their behavior, effectively building a physical and chemical fortress around the weed to protect it from the poison.
The tumor is literally conscripting healthy human tissue into its own defense militia.
Wow.
And that ecosystem dynamic makes treating the disease infinitely more complicated. because the resistance isn't just a property of the cancer cell anymore. The persistence is an emergent property of the entire corrupted neighborhood.
So developing a single biomarker or a single drug to overcome that level of cooperation is just a monumental task.
It's massive, yeah.
But, the Alonso study didn't just point out the problem. They actually found a potential vulnerability in this fortress, didn't they? Like, if the tumor absolutely requires this massive neighborhood-wide inflammatory response to survive the initial drug strike, is there a way to weaponize that knowledge?
Oh, absolutely. That was the next logical step in their research. They identified that this early inflammatory phase isn't just like a side effect. It is a structural load-bearing pillar of the tumor's survival strategy.
So they decided to try and demolish that pillar.
Exactly.
But how do you stop a microscopic fire alarm?
Well, they zeroed in on a specific protein called tank-binding kinase 1, or TBK1.
Okay, TBK1. And a kinase is essentially a biological switch that turns other proteins on or off, right?
Correct. In this context, TBK1 acts as a master switch that drives the survival-based inflammatory response. So to test this, the researchers used patient-derived organoids.
Oh, organoids. Those are incredible tools.
They really are. They're essentially miniature, three-dimensional versions of a patient's actual tumor grown in a laboratory dish. They preserve the exact spatial architecture of the original cancer.
That is so cool.
It is. So the researchers treated these organoids with a combination approach. They administered the KRAS inhibitor to shut down the main engine and simultaneously added a drug that inhibits TBK1.
Shutting down the cancer mutation while simultaneously cutting the wire to the fire alarm. What was the outcome?
The results were powerfully synergistic.
Synergistic meaning the combined effect of the two drugs was vastly greater than just adding their individual effects together, right?
Exactly. By inhibiting TBK1, they suppressed the tumor's ability to trigger that crucial inflammatory adaptation. They completely pulled the rug out from under it.
The implications of that are just staggering. I mean, it suggests that if we intervene early enough, if we block that TBK1 pathway right as we deliver the KRAS inhibitor, we might trap the tumor. We prevent it from ever reaching that highly resistant dual-state progression phase.
It represents a fundamental shift in strategy. Instead of just targeting the initial genetic mutation, we are proactively targeting the tumor's biological adaptation. We're anticipating its next move.
Okay, let's do a final synthesis of these mechanics before we look at the real-world application. We've learned that resistance is spatial tumors organized into specialized neighborhoods. We learned that tumors weaponize their environment, forcing healthy stroma and immune cells to build an inflammatory fortress. And crucially, we learned that if we can identify the master switch of that fortress, like TBK1, we can cut off the cancer's escape route. Which brings us to the most urgent question for you, the listener. How does this bleeding-edge science actually reach a patient in a hospital today? How do oncologists see these invisible, shape-shifting neighborhoods?
Yeah, that question highlights a massive diagnostic dilemma. There is this profound gap between the high-resolution maps we can build in a research lab and the tools available in routine clinical practice right now.
Right.
Currently, to determine if a colorectal cancer treatment is working, oncologists rely almost entirely on gross radiological assessments. We use standard CT or MRI scans.
But a CT scan is essentially just an anatomical photograph. It shows you the physical volume of a mass. It just tells you if a lump grew 3 millimeters or shrank 3 millimeters.
And that is exactly the problem. A scan offers absolutely zero functional insight into the internal biology of that mass. It cannot tell an oncologist if the cells inside have covertly shifted from a rapid growth fetal state to a highly defensive Paneth cell state.
So by the time a tumor visibly grows on a scan, the biological battle has already been lost.
Exactly.
Well, the obvious answer seems to be a biopsy, right? Like, if the scan can't see the cell state, just use a needle to extract a tissue sample and run that incredible spatial transcriptomic analysis we talked about earlier.
I mean, in a theoretical world, yes. Performing serial biopsies every few weeks would allow us to perfectly track the tumor's adaptation. But clinically, repeatedly driving biopsy needles deep into a patient's visceral organs carries very real severe risks of bleeding and infection.
That makes sense. It's too invasive.
It is impossible to justify that risk just for routine monitoring.
But beyond the physical danger to the patient, doesn't a needle biopsy suffer from a massive mathematical flaw here? It's kind of the classic blind men describing an elephant problem.
Oh, perfectly said. That is the exact limitation caused by the spatial organization we uncovered. If a tumor's resistance mechanisms are geographically segregated, a needle is only going to sample a tiny, tiny fraction of the mass.
Right, so you might extract cells from a neighborhood utilizing the fetal resistance strategy and completely miss a neighborhood mere millimeters away that is relying on the inflammatory strategy.
Exactly. A single needle biopsy fundamentally masks the true, terrifying diversity of the entire tumor ecosystem.
Okay, so if scans are blind to biology and biopsies are dangerous and inaccurate, what is the roadmap forward? How do we monitor a shape-shifting tumor in real time?
Well, the oncology field is investing heavily in non-invasive circulating biomarkers, primarily through analyzing cell-free DNA or cfDNA.
Cell-free DNA.
Yeah. As tumor cells grow, turn over, and die, they shed minuscule fragments of their genetic material directly into the patient's bloodstream. So by drawing a simple vial of blood, we can sequence that shed DNA.
But wait, earlier we established that the tumor isn't just acquiring new DNA mutations, right? It's changing its epigenetic software, its cell state. Can a blood test actually detect a software change?
And that is the true frontier of this technology. We are now developing epigenetic phenotyping of blood.
Okay, what does that mean?
We aren't just reading the letters of the shed DNA. We are looking for highly specific chemical tags. They're called methylation marks attached to the DNA fragment.
Oh, like microscopic Post-it notes.
I love that. Yes, exactly, like Post-it notes that reveal what state the cell was in before it died. They could theoretically tell us if a shed fragment came from a cell acting like a fetal cell versus one acting like an inflammatory Paneth cell.
That sounds like science fiction becoming reality. But I imagine interpreting that data is incredibly complex. I mean, if a tumor has multiple distinct neighborhoods, all shedding different DNA tags into the bloodstream simultaneously, the blood sample just becomes a chaotic, blended smoothie again.
And that is the major cautionary note the authors of the paper emphasize. Advanced blood tests are powerful, but they will struggle to definitively untangle multiple, spatially distinct, co-evolving resistance mechanisms.
Right, because if you see both fetal and inflammatory markers in the blood, you don't know if they're cooperating in the same neighborhood or fighting entirely different battles on opposite sides of the tumor.
Exactly. So the ultimate solution will likely require a multimodal approach, integrating these high-resolution blood tests with next-generation phenotypic imaging. So, scans that can actually visualize biological activity, and perhaps occasional, highly targeted tissue sampling when absolutely necessary.
That makes sense.
Only by combining these tools can we build a fully personalized dynamic map of a patient's exact resistance state.
It is an incredibly complex puzzle, but I mean, the pieces are finally starting to fit together. Let's look at the big picture of everything we've explored today. We started with KRAS inhibitors, the supposed holy grail of targeted cancer therapy, but we discovered that colorectal cancer refuses to be a static target.
Right.
It fights back not merely by mutating its genetic code, but by fundamentally transforming its cellular identity. It shifts from rapid expansion into a deeply defensive stem cell state. It spatially organizes into distinct, cooperating neighborhoods, and it forcefully recruits its surrounding healthy tissue into a massive inflammatory riot.
It really is a marvel of biology, even if it's a deadly one.
It is. Yet, by mapping this incredibly complex cartography, researchers have uncovered new Achilles heels, like the TBK1 pathway, proving that even a shape-shifting fortress has structural vulnerabilities.
It really forces a profound philosophical shift in how we approach the disease. And, I want to leave you with a final concept to consider. For over a century, oncology has largely treated tumors as chaotic, rapidly dividing mistakes. We built our treatments around finding a bigger hammer to smash the fastest-growing cells.
Just brute force.
Right, brute force. But the science we discussed today reveals that tumors act more like highly organized, supremely adaptive ecosystems that constantly communicate with their environment. And if that is true, then the future of oncology cannot simply be about reacting to the tumor we see today.
It has to be predictive.
Exactly. It's about anticipating the tumor's next evolutionary move on the map and barricading the road before the cancer even realizes it needs to turn.
What a brilliant paradigm shift. We basically stop playing a reactive game of whack-a-mole and start outsmarting the tumor it is actively trying to become. Well, thank you for joining us on this deep exploration of the cellular frontier. We encourage you to keep questioning the world around you and look past the obvious surface explanations. Because as we've seen today, some of the most profound mysteries and the most complex evolutionary battles are happening microscopically right inside the living blueprint of our own cells.